Abiotrophia defectiva. General information. Abiotrophia defectiva is a fastidious organism that requires a complex medium enriched with L-cysteine or vitamin B6 as well as other unique nutritional requirements that are essential for growth. Taxonomy of Abiotrophia defectiva. Family. Ericaceae genus. Abiotrophia and Granulocytella formally. Streptococcus defective. Nutritionally variant Streptococci or satelliting Streptococci. Natural habitat of Abiotrophia defectiva. They are members of normal flora of the oral cavity, or upper respiratory tract, as well as the intestinal tract. Clinical significance of Abiotrophia defectiva. They are opportunistic pathogens, and are formally known as NVS. Nutritionally variant Streptococci are normal residents of the oral cavity, and are recognized as agents of endocarditis involving both native and prosthetic valves. These organisms have also been isolated from other types of infections, including ophthalmic infections, peritonitis in patients undergoing continuous ambulatory peritoneal dialysis, musculoskeletal infection, septic arthritis, and a breast implant-associated infection. Although it is rare for Abiotrophia defectiva to cause endocarditis, they have a greater morbidity and mortality than endocarditis caused by other Streptococci, due to its poor response to many antibiotics. Cases are rare. Gram stain. The gram stain of Abiotrophia defectiva typically reveals gram-positive coccobacilli, which are small, oval-shaped bacteria measuring approximately 0.4 to 0.5 micrometers in size. These bacteria are often arranged in characteristic pairs or chains, resembling the typical appearance of Streptococci. However, it's important to note that the microscopic morphology of Abiotrophia defectiva can exhibit some variability depending on the growth medium used. In some cases, the bacteria may appear gram-variable or exhibit a pleomorphic morphology, meaning they display a range of shapes and sizes, sometimes appearing enlarged and irregularly shaped. This morphological variability can sometimes pose challenges in identification, highlighting the importance of considering growth conditions when interpreting gram stain results for Abiotrophia defectiva. Culture characteristics. Abiotrophia defectiva is a facultative anaerobe, which means it can grow in both the presence and absence of oxygen. The bacterium does not exhibit growth on blood agar or McConkey agar. However, it can grow on chocolate agar, forming small alpha hemolytic colonies with a maximum diameter of 0.2 millimeters. Additionally, it demonstrates the ability to grow as satellite colonies adjacent to Staphylococcus aureus or other Staphylococcus species. These satellite colonies can be either non-hemolytic or alpha hemolytic. Lastly, Abiotrophia defectiva is capable of growth on brucella blood agar with vitamin K and hemidin, BBAO. Characteristics. Abiotrophia defectiva displays several key characteristics. It is a gram-positive bacterium, meaning it retains the crystal violet stain during gram staining. The bacteria are cocci, or spherical in shape, and often arrange in chains, a characteristic of Streptococci. A defectiva exhibits both aerobic and anaerobic growth, showcasing its adaptability to different oxygen environments. However, it fails to grow on standard blood agar or McConkey agar, indicating its fastidious nature and specific growth requirements. The bacterium exhibits alpha hemolysis, causing a partial breakdown of red blood cells resulting in a greenish discoloration on blood agar. Biochemically, it is catalase negative and oxidase negative, meaning it doesn't produce these enzymes. It is also purase positive, non-modal, and susceptible to vancomycin but resistant to colistin. Notably, A. defectiva displays a satellite phenomenon, requiring the presence of other bacteria for growth, and it is lipase positive, indicating the ability to produce the enzyme lipase. Poem. In the realm of microbes, a tale unfolds, Abiotrophia defectiva, its story untold. A fastidious being, with needs so refined, L-cysteine or B6, its growth intertwined. Once known as Streptococcus, in names it did dwell, nutritionally variant, its secrets to tell. In oral and respiratory realms, it takes its stand, a member of the flora, across the land. Opportunistic pathogen, in shadows it hides, endocarditis its mark, where danger resides. native or prosthetic, valves under attack, morbidity and mortality, leaving its track. Gram-positive cocobacilli, in pairs and chains, microscopic morphology, its form explains. On blood agar, no growth it displays, chocolate agar's embrace, its presence conveys. Satellites it forms, near Staphylococcus's mite, non-hemolytic or alpha, in dim morning light. Facultative anaerobe, in air or its lack, biochemical traits, its identity to unpack. Catalase negative, oxidase it denies, purase positive, its nature implies. Non-modal and susceptible, to vancomycin's call, colistin's resistance, standing tall. A rare cause of endocarditis, its impact profound, in the microbial world, its presence is found. Abiotrophia defectiva, its secret we glean, a fascinating organism, in the grand scheme.